Hello guys, you're welcome to Alicia's Med Hub. Today we'll be discussing genetic disorders or hereditary disorders. You can call it hereditary disorders. Same thing. So hereditary disorders or genetic disorder by definitions are derived from one's parent and they are transmitted in the germ layer through generations, therefore are familiar, right? By definition, they are derived from one's parents, that is, they are gotten from your parents, right? And they are transmitted in the germline through that is through the reproductive cells, right, through the generations, therefore are familiar. They are familiar because they are gotten through the family. So congenital simply implies born with. Some congenital diseases are not genetic. Some congenital diseases are not genetic. For example, congenital syphilis. And not all genetic diseases are congenital. Well, okay, individuals with Huntington's disease, for example, begin to manifest their condition only after 20 years or 30 years. What this statement is saying is that there are certain disease, right, that are not, you are born with them, but they have no connection with your genes, right? For instance, congenital syphilis. Syphilis is an infection. It's an infection, right? Just like gonorrhea, gotten through uh, mostly through sexual intercourse, right? So now, if the mother has syphilis, most likely, um, if the disease is not treated properly, most likely the child is going to come down with syphilis. So that is called congenital syphilis. However, it has no genetic basis, right? It has no genetic basis. No, it doesn't come through germline, right? Has nothing to do with the genetics of the mother and the baby. Now, no, of course, not all genetic diseases are congenital as well. Not all genetic diseases are congenital. That is, there are certain genetic conditions that do not manifest at birth. You understand? You're not born with them; they are there. However, they require they require a space of time to manifest effectively. And a very good example is Huntington disease. So it manifests, during your 20s or your 30s. So this is Turner syndrome. It's a genetic condition, right? You could see the web neck. You could see the widely, displaced nipple, broad chest, right? Now this is a female. Yeah. So this is Turner syndrome. This condition has genetic basis. So this is a hereditary disorder. It's a genetic disorder. This is Down syndrome. You could see the moon face shape, right? Moon face shape, typical for Down syndrome babies. They usually have mental retardation, slow growth, and all of that, right? So now, genetics is the study of single or a few genes and their phenotypic effects. Genomics is the study of all the genes in the genome and their interactions. Genome is a complete set of chromosomes derived from one part. The haploid number of a gamete and the human genome sequence is complete. Human genetic architecture slash introduction. Less than 2% of the human genome encodes protein, whereas more than one half represents blocks of a repetitive DNA sequence whose function remain mysterious. 20,000 to 25,000 gene codes for protein rather than 100,000 predicted. So initially 100,000 were predicted that 100,000 genes were predicted for protein synthesis. However, in recent studies, it has been discovered that 20 to 25,000 genes codes for protein rather than actually 100,000 that been predicted initially. So, quite remarkably, this figure is about the same as that of the mustard plant with 26,000 genes. Right? So, you're just making the comparison here. By alternative splicing, the 25,000 human genes can give rise to greater than 100,000 protein. Splicing, this is the reaction that removes introns and join extrons together, resulting in mature messenger RNA. Now, during protein synthesis, there is a section or there is a point or junction where introns, are spliced out and extrons, right, and extrons are fused together. All these things are carried out by enzymatic action. So DNA microarray analysis of tumor is an excellent example of genomics in current clinical use. More than 99.5% DNA sequence are shared by any two individuals. More than 99.5% of DNA sequence are shared by any two individuals. Less than 0.5% equals the DNA responsible for whole phenotypic variations. The secret to disease predisposition and response to environmental agents and drugs must therefore reside within these variations. This 0.5 represents about 15 million base pairs. Significant, so significant. So little but so significant. Now, the two most common forms of DNA variations in human genome are single nucleotide polymorphism and copy number variations. The two most common forms of DNA variations in human genome are single nucleotide polymorphism and copy number variations. Single nucleotide polymorphism represents variations at single isolated nucleotide positions and are almost always biallelic. Therefore, one of the only two choices exists at a given site within the population, such as um, adenine or thymine. Single nucleotide polymorphisms may occur anywhere in the genome within the exons, introns, or the intergenic regions, but less than 1% of the single nucleotide polymorphism occur in coding regions. So copy number variations are recently identified types of genetic variations made up of variable numbers of large contiguous stretches of DNA ranging from 1,000 base pairs to millions of base pairs. Their loci share some similarities with single nucleotide polymorphism, biallelic, and this can be found simply duplicated or deleted in a subset of the population. In other instances, they are complex rearrangements of genomic material with multiple alleles in the human population. It is believed that copy number variations are responsible for 5 to 25 million base pairs of sequence difference between any two individuals. It is believed that copy number variations are responsible for 5 to 24 million base pairs of sequence difference between any two individuals. So thank you for listening. Please kindly like, subscribe, and share if you find this educative. Thank you.